Well, Michael, thank you for meeting with me.
My pleasure.
So why don't you tell us a little bit about who you are? Where do you come from?
Sure.
How'd you end up in Utah?
Sure. So I'm a professor of biochemistry in the School of Medicine here at the University of Utah. I'm originally from Miami, Florida, and been bouncing all around the country for different levels of training. I'm an MD PhD by background, but decided to go fully into research instead of pursuing residency training. My lab is pursuing studies of mirror image proteins and peptides with diverse therapeutic applications.
Cool. Do you want to tell us what a mirror image protein is?
Sure. So just like your hands, there's left and right-handed versions of proteins. And for reasons that no one really understands, nature picked the left-handed versions early in evolution, and all life has standardized on that ever since. And so in theory, there could be right-handed life and right-handed proteins, but they're not found in nature. And so we make them by chemical synthesis, and we're excited about them because they're not recognized by natural life. They're not processed, degraded, and so in theory, they can be used as drugs that would have very, very long lifetimes. It can be administered at much lower cost, with lower frequency than natural drugs, which do get metabolized by the body.
So this is kind of a useful tool to make all kinds of different drugs. Do you want to tell me some applications of these drugs, like specific targets you've gone after?
Sure. So I'm a virologist by training, and so my early area of interest in applying these mirror image peptides was to virology, particularly to HIV. And this is a project we've been working on for over 10 years, and we have these mirror image peptides that we've designed and optimized over the years, and are very effective inhibitors that block the HIV entry process, basically preventing HIV from getting access into a human cell. And so we're now pursuing preclinical studies to see if these compounds can be used to prevent and treat HIV.
And how are those going?
They're going very well. So these compounds look very, very effective in tissue culture models of HIV. We're now doing animal models with collaborators, and those have been going very well also. And so we're now hoping to advance to human clinical trials in the next year or so with these compounds to see if they work in humans as well as is predicted by the animal studies and the lab studies. Very exciting.
Yeah, that's really exciting stuff. Do you want to tell us some more projects you have in the works, if it's not too confidential?
Sure. So HIV was our first project, and we've had a lot of success there. And we now want to see, can we broaden that out and apply it to other systems? And so we're very interested in working on other viruses that have a similar way of getting into cells. And so we've been adapting our mirror image drug discovery platform to see if that works against other viruses, and it looks like it will. And so we have earlier stages of inhibitors that are being developed against viruses like Ebola that we think would also be incredibly useful as a tool to prevent or treat viral infection. And then we're looking even broader to look at other targets outside of virology to see how broadly we can use dipeptides against bacterial pathogens, as well as targets in humans like cancer targets, inflammation targets, things like that. And so we're at the early stages of that, but our big focus has been on developing the kind of technical background and the chemical synthesis tools that are necessary to make these larger, more complex targets and put them into our mirror image drug discovery platform.
So what are some challenges of making these large dipeptides?
The challenges are that it's difficult to know when you make a large mirror image protein whether it's functional or not, because you can't tell using traditional functional techniques. Let's say you have an enzyme. If it's mirror image, you have to provide it mirror image substrates in order to find out if it's actually active, and that can be quite challenging. So we have to use a lot of indirect methods that look at the overall structure of the protein and some of its behavior to see that it looks like it's identical to the natural protein, except that it's in the mirror image. So for instance, we can look at the size of a protein and show that it's very similar or identical to the natural version of a protein. That's a good sign, showing that it's folded properly. We use techniques like circular dichroism to measure the secondary structure of a protein. And again, we can see that we have the same secondary structure in the natural and the mirror image protein. That's a good sign that it's folded properly and that it is the authentic protein except in mirror image.
Cool. So it sounds like you've got a lot of different things in the works. I think that'll probably do it for us today, but thank you so much for meeting with us.
Sure, thank you very much.
All right.